First of all, I would like to thank the organizer for making this conference happen and in giving me this opportunity to present my work. My name is Behzad Iravani, and I'm a PhD student at the Department of Clinical Neuroscience, Karolinska Institute. And the title of my talk today is Early Processing of Odor Valence in the Human Olfactory Bulb. Olfactory bulb is the first processing stage of olfaction in central nervous system. There are tons of animal study on olfactory bulb in which they could link it to the long list of olfactory tasks like odor discrimination, concentration invariant recognition, odor segmentation, and odor pattern recognition. However, we know very little about human olfactory bulb, mainly due to the lack of non-invasive methods to measure the function of human olfactory bulb. Here you can see common neuroimaging techniques and their shortcomings when it comes to measure the function of human olfactory bulb. Recently, we developed a non-invasive recording that can assess the function of human olfactory bulb. In our method, we use a little bit different configuration of the EG electrodes, and we combine it with a source reconstruction algorithm to actually capture the olfactory bulb response on a scalp level. Our method has recently published in Nature Communication. Here you can see the summary of our method. So in addition to the 64 EEG electrodes, we added 4 extra electrodes, and these are active electrodes on the forehead, and we call them electrobulbogram or EBG. So the algorithm, of course, starts with the data collection, artifact detection and correction, and epoching. In parallel, we also collect the data from the positions of electrodes and digitalize them to use them in a source model. The analysis happened on a 2 levels, sensor level and source level. And for head modeling, we consider spherical models. And we include 4 tissues: a skull, scalp, gray matter, and white matter. And for source modeling, we consider undetermined sources, meaning that we have dipoles everywhere in the brain that has a gray matter density more than 40%. Using our method, when we assess the time frequency response, we found results similar to that of animal literature and intracranial data. On this graph here, the horizontal axis is time and the vertical axis is frequency. And the power of the oscillations is color-coded. So shortly after the odor onset, we see a gamma synchronization that is induced by odor, but not air. Later on, using a source reconstruction method, we could localize this synchronization onto the olfactory bulb. We also assessed the test-retest reliability of the measure on a separate data set. So we collected the data from 19 healthy individuals over 3 sessions, and we looked at how reliable the signal is, where we found the medium effect size and also high correlation between the sessions. Moreover, we also see that we have a low dispersion rate determined by meta-regression. So all in all, we conclude that the measure has a good reliability, and it's comparable, for example, to the ERP results. Alongside the gamma oscillations, animal literature showed that slower oscillations, for example, beta, also has a major role in olfactory processing in animals. We also assessed this in our data by looking at the time resolved phase amplitude coupling, where we found a strong coupling around 250 to 500 milliseconds after the odor onset. We looked at this closer using a comodulogram, where the horizontal axis on this graph here is the frequency of low oscillations, and the vertical axis is the frequency of high oscillations. We realized that this coupling appears in a narrow-banded low frequency close to the mu oscillations. The exact functional role of human olfactory bulb in perception of odor is not yet clear. There are potential dimensions of odor that olfactory bulb can process, like intensity, valence, familiarity. Today we focus on valence. Based on the past studies, it seems that valence is the primary dimension of olfactory bulb, that olfactory bulb process. Moreover, it can be linked to the approach avoidance behavior from animal literature. Generally, it seems that olfactory bulb helps making decision on approaching avoidance behavior, and also particularly for a negative stimuli. Here, the advantage here we have with a human is that we can easily ask them about the perceived valence, whereas this is not possible with the animal literature. Given the early gamma synchronization followed by a strong coupling with the slower oscillation, next we ask if olfactory bulb facilitates area avoidance response. Based on the past literature, we know that mu desynchronization is a good measure of preparatory motor response. So based on the timing we found in olfactory bulb and considering a hypothetical reaction time, we expect to see this preparatory avoidance response around 300 milliseconds. We assessed this in our data using a mixed effect models, where we looked at the power of mu desynchronization and valence for the time point from 300 to 400 over the whole scalp, where we found a strong association between this desynchronization, the mu desynchronization and valence over the motor cortex. In the next experiment, we assess whether this preparatory mu desynchronization would elicit any actual avoidance response, motor actions. Since we wanted to look at this on a multiple time windows and to avoid multiple comparison problem, first we did a pilot experiment, which later on we tried to replicate our results from the pilot experiment in the main experiment with a bigger sample size. Here you can see the setup. We ask our participants to stand on a force plate facing a wall with a fixation cross on it. And we, using a respirometry and olfactometer, we gave our participants either pleasant or unpleasant odors in phase with inhalation. And we also collected the balance data and body sway measures using a force plate. And this is what we found. On this graph, the vertical axis is posterior-anterior movement, which the negative values here can be interpreted as backward motion, and the positive values can be interpreted as forward motion. So in line with the timing of mu desynchronization, we see around 500 millisecond backward motion or avoidance response for unpleasant but not pleasant odors. So this avoidance response can be linked to the olfactory bulb with time. Later on, we could replicate this result on a main experiment with a larger data sample size, both in a frequentist and Bayesian framework. We get a base factor large enough that we could accept the hypothesis and reject the null. In conclusion, the EBG measure can be used to assess relationship between the olfactory bulb signal and behavior. We also found that phase amplitude coupling appears between gamma and slower oscillations in human olfactory bulb. Therefore, our results suggest that slow oscillations in olfactory bulb facilitates avoidance response to odors. Thank you very much for your attention.
Hello, everyone. This is the topic of my talk today. Let's start with a brief intro of our introduction. There are discrete patches throughout the visual cortex processing different object categories such as faces, bodies, and different scenes. There has been an important question in the literature asking whether these areas are developed through the effects of genetic or environment or the combination of both. And if a combination of both, how much is the strength of genetic and environmental influences on the organization of these areas? There are several studies in favor of either genetic or environmental influences. However, the results are conflicting and there is no consensus over one unique answer. So to answer this question, we conducted a twin study claiming that if we found more similarity in identical twins, this would suggest a strong role for genetic because as we know, identical twins share 100% of their genetic information. However, non-identical twins share only half of their genetic information. So for performing this study, we took advantage of the twin data from Human Connectome Project database that includes 134 monozygotic twins and 78 dizygotic twins, which I may refer to them as MZ and DZ. And we included the structural and functional imaging which were all fully analyzed by the HCP pipeline. The task that we were interested in was a working memory task. Blocks of faces, bodies, places, and tools were presented to the subjects, and subjects were doing either a 2-back or 0-back task. If we compare activation pattern of a specific category with all other categories and collapse them across the memory conditions, this could be used as a functional localizer to detect the category selective areas. Thereby, we will have an activation pattern for each category based on 3 contrasts that I've listed here. And finally, our maps were based on z-statistics maps for each contrast in each subject. As the first step, we did a whole brain analysis in order to assess whether the activation maps are more similar in identical twins compared to non-identical twins by performing intra-class correlation analysis between a twin and the co-twin of a pair in MZ and DZ twins. We then visualized the intra-class correlation coefficients on 2D flat maps of the brain. And as you can see here, the correlation coefficients are higher in MZ group compared to DZ, reflecting more similarity in the activation pattern of MZ twins, particularly on the posterior part of the brain. To quantify this effect, we performed another analysis, which was a Pearson correlation test between a twin and the co-twin, and plotted the distribution of correlation coefficients for all MZ and DZ twin pairs in all 3 categories. And as you can see again, MZ twins showed higher correlation coefficients on average compared to DZ twins. Relatedly, we did similar analysis, but this time dividing the brain into 4 cortical lobes, including occipital, parietal, temporal, and frontal lobe, to study them separately. And we plotted the mean correlation coefficients for MZ and DZ across all the categories. And as you can see here, the findings that we found previously is only evident on the occipital lobe, which is consistent with our ICC maps that we saw earlier. So here comes this question that, are these category-related activation genetically determined equally throughout the visual cortex, or are the genetic effects stronger in dominant-specific, or may I say more selective areas? To answer this question, we applied a range of thresholds on the Z maps, binarized the maps, and to calculate the spatial overlap between a map of a twin and the co-twin in MZ and DZ. We then plotted the mean spatial overlap of all pairs in MZ and DZ across the range of the thresholds that we had. And the higher the threshold is, the more specific the regions are. So as you can see here, by increasing the thresholds, we see more separation between the MZ and DZ twins. So taking all these results together, we can say that there is more similarity in the activation pattern of MZ twins compared to DZ twins, particularly on most selective regions. Thereby, we decided to focus more specifically on category selective areas for the rest of our analysis. And for that, we extracted top 1% of voxels, which had the highest Z values in a given contrast from the group average activation map of 787 subjects of the HCP database. And we visualized them here. And as you can see, The patches that we extracted are nicely correspondent to the previously known patches in the literature, such as FFA and OFA for face areas, and etc. So to answer clearly our primary question, we performed a more sophisticated approach, which was called structural equation modeling, which enabled us to decompose the genetic and environmental components, and thereby we could detect the voxels that are highly influenced by the effect of genetic. We then visualize these voxels here in red, you can see them. And these are the voxels that are highly influenced by the genetic components. And we can say that a considerable number of genetic voxels was observed in almost all of these patches. However, the genetic influence was heterogeneous within these areas, and only part of these patches showed the genetic voxels. So we thought that maybe these genetic, non-genetic patches are related to some distinct structural properties. And to investigate that, we investigated the relationship between genetic effect in category selective areas and structural properties of cortical gray matter by calculating the cortical curvature, thickness, myelination in genetic and non-genetic voxels of all these categories. Interestingly, we found that genetic face voxels had higher cortical curvature, thickness, and myelination compared to non-genetic face voxels. In contrast, genetic place voxels had higher cortical curvature but lower cortical thickness and myelination compared to non-genetic place voxels. So finally, to address the concern that maybe the similarity that we see in function is related to the similarity in structure in identical twins, or in other words, whatever we found so far is related to the similarity in anatomy. We looked into the ICC maps of the thickness, curvature, and myelination. And as you can see here, again, we can say that the correlation coefficients are higher in MZ twins compared to DZ twins, but the pattern of similarity is completely different from what we had already seen for the functional maps. Moreover, we performed another analysis, which was structural equation modeling for curvature, thickness, and myelination to detect the genetic voxels of these structural features, and showed them here in green, while red represent the genetic voxels of functional activation. And as you can see here, there is literally no or few overlap between them. So we can say that the similarity that we saw in the function cannot be explained by the similarity in the structure. And what we found before in the functional activation is independent from the similarity of anatomy in MZ twins. So finally, to conclude, the comparison between MZ and DZ twins in this study revealed the heritability of category selective areas in visual cortex. This genetic influence was not homogeneous across these areas, and only part of these areas showed significant genetic effect. These genetic subregions within face and place areas contain specific structural properties, such as genetic face and place voxels were more prevalently found in regions with higher cortical folding, which can be explained by a tension-based theory of cortical morphogenesis, assuming that these voxels are strongly interconnected. The mechanical tensions along axons of these connections would force the cortical tissue to get folded. Moreover, an increased cortical thickness in genetic face voxels suggests that these voxels may contain a higher number of neurons, perhaps for a sparse coding of faces. While myelin content has also been shown to be inversely correlated with intracortical circuit complexity, thereby a decreased cortical myelination in genetic place voxels suggests that these voxels may have more dynamic intracortical circuits, perhaps for a distributed coding of places. Thank you all for listening.
Thank you for organizing this session. I'm gonna talk about neurophysiological sources of multimodal neuroimaging in humans. The aim of this study is to investigate the extent to which the spatiotemporal dynamics of brain activity obtained by EEG or fMRI or the similarity-based EEG fMRI fusion are consistent with that of neural activity obtained by ECOG data. EEG and fMRI are two major non-invasive methods for imaging human brain function. EEG is known for its excellent temporal resolution but poor spatial resolution, whereas fMRI has a good spatial but poor temporal resolution. ECOG is an invasive technique providing simultaneous spatiotemporal mapping of neural responses. So it gives the possibility to compare neural dynamics with the results obtained by EEG fMRI fusion, but with the limitation of not having the whole brain coverage. In this study, we recorded EEG and fMRI data separately from 21 healthy volunteers. The stimulus set included 125 images from the animal, chairs, face, fruit, and vehicle categories. There are 5 identities in each category, and the identities are presented in 5 different sizes or orientations. This is the exact stimulus set that is used to record ECOG data by Liu and colleagues in the Kramer lab at the Harvard Medical School in 2009. The experimental parameters are also designed similar to that of ECOG data. In the following slides, I will present the results for the fMRI and ECOG comparison. To compare ECOG and fMRI data, we extracted brain visual regions in occipital inferior, fusiform, lingual, parahippocampal, temporal inferior, and pole temporal. Then, for each subject and each region, we extracted condition-specific voxel activity patterns. These are the inputs for SVM classifier to examine the category selectivity and the robustness to the scale and orientation changes in these regions. The results for ECOG and fMRI data are shown here. For each region, there are 5 bar charts, each showing the accuracy for classifying each category versus the all other categories. The bars are color-coded. For example, the blue bars are the accuracy for discriminating between the faces and all other categories. As shown in this figure, occipital inferior and fusiform areas are more selective to the faces than to the other categories in ECOG and fMRI. For parahippocampal and temporal inferior, fMRI and ECOG patterns of selectivity are different. fMRI responses show higher selectivity to the vehicles, whereas ECOG responses are more selective to the faces. To examine scale invariance, we trained the classifier with the images in the size of 3-degree visual angle and tested by the resized one. This is the average classification performance across different scales. We see that category information can be read out robust to the face changes in occipital, inferior, and fusiform in both fMRI and ECOG data. None of the lingual, parahippocampal, IT, and pole temporal regions show significant decoding accuracy across different scales in fMRI. However, in ECOG, these regions are more selective to the faces or fruits. A similar analysis was performed to investigate category selectivity under orientation changes. The classifier is trained using the images in the orientation of 0 degree and tested by the rotated images. We observed that the classification performance is higher for faces in the fMRI and ECOG in fusiform area. In ECOG, the accuracies for faces and fruits are almost identical in occipital inferior, whereas in fMRI, there is a significant difference between these categories' performances. In the next part, the results for ECOG and EEG data comparison will be presented. In order to evaluate the earliest time point at which we could reliably decode category information, we constructed condition-specific vectors using the EEG amplitudes over all electrodes and use SVM classifier to decode object categories at each time point. In this figure, we see that the classification performance, as a function of time, averaged over all categories and subjects. The latencies and performance patterns for EEG and ECOG are compatible. According to this analysis, human brain decode category information as early as 100 milliseconds post-stimulus onset. The bar charts show the range of the classification performance for categories. For both modalities, the performance is higher for the face category. The third section is combining EEG and fMRI data and comparing the results with the ECOG results. For fMRI, in 6 aforementioned ROIs, we extracted condition-specific voxel activation values yielding 125 pattern vectors. Then we calculated the pairwise dissimilarity for all experimental conditions. The dissimilarity measures are assigned to a 125 by 125 fMRI dissimilarity matrix. Similar analysis was conducted on each data to obtain representational dissimilarity matrix at each time point. And then for each region, we correlated fMRI RDMs to EEG RDMs at each time point, and this gives signal over time. The first time point at which the correlation of the similarities is significant is comparable to the mean latency in ECOG data. There is a similar latency in occipital, inferior, fusiform, and lingual areas, but not in parahippocampal, temporal, inferior, and pole temporal. In some, spatial analysis showed a great consistency between ECOG and fMRI results in occipital, inferior, and fusiform areas according to the time domain analysis, and regardless of the location, human brain can decode category information after 100 millisecond stimulus presentation. There is more temporal similarity between EEG and ECOG patterns compared to the spatial similarity between fMRI and ECOG.
So, hello, I'm Stefania Mattioni, and today I will speak about how the onset of blindness might affect the interplay between cross-modal and intramodal plasticity. In all the activity that we do in our everyday life normally activate our 5 senses. And all this information are then processed by the brain. And the classical view of brain functioning suggests that each region, different region in the brain will actually analyze the different modalities separately. For example, the temporal cortex will analyze the auditory information, while the occipital cortex will analyze the visual information. But is this division really unchangeable? Study on early blindness actually suggests that this is not the case. In fact, in this population, the occipital cortex, now deprived from its visual input, gets invaded by other modalities, such as the audition, and it starts to process auditory information. This phenomenon is known as cross-modal plasticity, and it has been reported in many studies in the past. And a more recent study also showed that even if the modality switches, actually the functional scaffolding of this region is preserved. For instance, we recently showed that the categorical representation in the ventral occipitotemporal cortex of VOTC is significantly similar in sighted for visual stimuli and in blind for sounds. Just to make an example, the region that prefers faces in sighted switches now and start to prefer voices in blind. So basically the modality switches, but the categories stay the same. However, there are still some crucial questions that are unanswered. First of all, if VOTC starts to process sound in blind, what happens in the temporal cortex, a region that we know is devoted to the same kind of process? And how the interplay between the occipital and the temporal region would change in blind people? And second question, how the onset of blindness can impact in the reorganization of the brain? To address these questions, we presented different sounds from 8 categories, include half animate, including neutral and emotional voices, and half inanimate, including birds and mammals. Birds and mammals in the animate, and then half inanimate, including tools and graspable objects, and then environmental and mechanical big sounds. I presented these stimuli in the fMRI to 4 different groups. So 2 groups of blind people, one of early blind and one of late blind. And each of these groups has its own age and matched sighted control group. Finally, I used the free surfer to select 2 regions of interest in each subject, the VOTC and the ventral occipitotemporal cortex and the temporal parcel. So using free surfer allowed us to work in the subject space. Okay, then what are our results? First of all, we used multivoxel pattern classification to look if VOTC and the temporal region were able to distinguish the different categories in our different groups. So I will start with the results from early blind and their sighted controls. As we reported also in our previous study, VOTC can decode the different categories significantly better than chance in both groups. However, we can see that the early blind, that is the light green here, have an enhanced accuracy compared to the sighted control in darker green. When we look at the temporal regions, also in this case, we see that these regions can decode the different categories significantly better than chance in both groups. But in this case, we see that the accuracy is actually enhanced in the sighted compared to the blind, driving to a significant group by region interaction. When we look at the result in the late blind, we see a similar profile, but in this case, some statistical differences are a bit milder. However, we also directly compare the early blind and the late blind after normalizing their data based on their own sighted controls. And in this graph then, that I will now show, the 0 values means that there is no difference from the sighted controls. What we found, as expected also based on the graph that I just showed you, is that in VOTC, we have in both groups positive values, meaning that both early and late blind have enhanced the accuracy compared to the sighted. In the temporal cortex, the situation is reversed. The values are negative. In fact, both blind groups have a decreased accuracy values compared to sighted. However, the important information that we should get from this graph is that there is no significant difference between the 2 groups of blind. Okay, so using this analysis, we cannot go further in the sense that we cannot really look at how each region represents the different categories in each group. To do that, we need another kind of analysis, the representational similarity analysis. Using RSA, we can in fact extract the dissimilarity matrices from each region and each group. And these matrices show how each category is represented compared to all the others in each region and group. The first question we wanted to address was whether the representation in VOTC and in the temporal parcel was more similar in the blind compared to the sighted controls, suggesting potentially an increased flow of information between these two regions in blind population. So what we did was to compute the correlation between each between the VOTC and the temporal matrices in each subject. And the result indeed show that in the early blind, the similarity of the representation between the VOTC and the temporal regions is significantly increased compared to the sighted. In the late blind, we have a similar profile, but this difference is not significant. However, when we directly compare the two groups of blind, once again, we don't find any significant difference between the two blind groups. So at least in the case of early blind, we can say that the similarity between the representation of VOTC and the temporal regions is increased compared to their sighted control. But this is actually not the only comparison that we can look at. There are many other comparisons between regions and groups that we could investigate. So to do that, we created a second-order dissimilarity matrix, including all these comparisons. For instance, the comparisons between the representation of VOTC in early blind with the representation of VOTC in the sighted controls. And then, in the same way, we included all the other possible comparisons. And we get this kind of matrix. To better understand the structure of this matrix, we applied a hierarchical clustering analysis on it. This analysis basically will divide the different parts of the matrix into two clusters according to their similarity. So basically, regions that have a more similar representation will be clustered together. Here I represent the clusters in two different colors. And as you can see, one cluster is basically made only of the VOTC of the sighted, while the second cluster includes both the temporal parcels of blind and sighted, and also the VOTC of the early blind, suggesting that the way VOTC represents the auditory categories in early blind is more similar to the way used by the temporal parcels, both in blind and in sighted, compared to the way used by the VOTC in the sighted. And interestingly, we see a very similar profile also in the late blind, in which we find exactly the same two clusters. So I really would like to share other further analysis that we did with you, but it's already time to jump to a quick summary. So our data suggests that early blindness induces a network-level reorganization in the processing of sound categories by concomitantly increasing the computational load of the occipital cortex and decreasing the computational load of the temporal regions compared to the sighted. Moreover, we observed an increased similarity in the representation of the occipital and temporal cortex in early blind, with the representation of the categories in the occipital cortex being more similar to the one in the temporal regions compared to the sighted. Finally, we show that the brain reorganization in late blind is overall similar to the one that we observed in the early blind, even though a bit less marked. This suggests that a late onset of blindness has not a qualitative impact on the brain reorganization, but might quantitatively reduce it. And with this, I thank you for your attention, and also my supervisor and my lab.
Good morning, everybody, or good night, depending on where you are on the planet. Today, I will talk about the fact that we are able to perceive a texture through different sensory sources, especially tactile information from our hands, of course, but not only, also auditory information related to our interaction on the surface. We can, for example, recognize the sound of crumpling paper. In this study, we try to find out whether textual representation in the brain are sensory specific and whether some audiohaptic interaction occur to improve textual perception. Until now, most of fMRI studies focused on haptic texture perception and stressed the importance of the secondary somatosensory cortex, the posterior insula, and strangely, activation of the posterior visual areas have been frequently reported during a pure tactile task, as well as during the pure visual task. This is what Stila and Satyan reported in their experiment. You can see here in blue the activation of the posterior insula and the postparietal operculum, plus the middle occipital cortex during the haptic texture exploration. And the middle occipital cortex was the same region also found activated during the visual texture perception. The interpretation of such activation in visual areas during texture haptic exploration is still debate, and its role remains unclear. Interestingly, far less is known about the influence of auditory cues on texture perception. Also, it has been shown a long time ago that the friction noise of fingers on the surface provides valuable information about the roughness of that surface. And there is one study, fMRI study, showing a specific activation of the less fusiform gyrus and the posterior superior temporal sulcus during an audio haptic matching task for object recognition. But we know almost nothing about brain region involved in audio haptic texture perception. So to address this question, we use a specific tactile device able to simulate virtual texture. Its principle is based on ultrasonic frequency vibration of the plate with very small amplitude that can modulate the friction parameter of the finger across the touchpad. This friction modulation can give rise to the participant the sensation that he is exploring a rather smooth or rough texture. And we use this tactile device in a psychophysical experiment, and we show that we can precisely estimate a discrimination threshold, and this precise tool is also able to detect the tactile impairments, like, for example, in older people. So we combined this tactile device, which was MR compatible, with a sound synthesizer, which produced two kinds of sound, a rather rough or smooth sound, to give, and this sound were produced in real time and modulate with the velocity of the finger movement of the participant on the touchpad, recorded through this optical sensor. So this movement sonification can give the participant's feeling that he has actually producing the sound by his own movement. So let me show you a video about the bimodal condition when the participants were asked to move their finger forth and back, while a smooth or rough sound was produced at the same time by their moving finger. Here is the full experimental procedure we performed during the fMRI acquisition. We had 5 runs, and for each run, it was composed by 4 blocks. One block was an haptic block, during which participants performed active movement with their right finger. Another block was the audio block, consisting in a passive listening to a texture sound. The rest block was basically a resting period during which the participant did nothing. And the bimodal block, consisting moving the finger during, at the same time, a perception of the sound, which was congruent with the texture explored by the subject. In 15% of the trials, participants were instructed to verbally report the roughness of the textures they perceived on a subjective scale, ranged from 1 to 10, with 1 corresponding to a very smooth texture and 10 a very rough texture. So speaking about the behavioral results, here you have the score reported by the participant, and you can see that they correctly classify the rough stimuli as rougher than the smooth stimuli. And in the congruent bimodal condition, you can see that the score were intermediate compared to the two unimodal score, meaning that in the bimodal condition, the two sensory information may have been integrated. What about the fMRI results? So we performed two types of analysis. A classical univariate analysis based on the general linear model with 6 conditions defined as regressor of interest, and several confounds extract from the denoising procedure implemented in TAPA's toolbox to account for non-neural noise and head movement related artifacts. We also performed an MVPA analysis consisting in a searchlight whole brain analysis to find brain region able to decode the texture. So to this end, we trained a classifier to identify the specific pattern of brain activity when presenting either smooth or rough texture. And this training was done on 3 out of the 5 runs, and then we test on the other 2 runs whether the classifier was able to decode the new smooth and rough trials with an accuracy above the chance level that was 50%. So first, let's have a look on the univariate results. During the haptic exploration of virtual texture with the haptic sensory information, the whole sensory motor network was, of course, involved, but also some frontoparietal region, the inferior frontal gyrus here and the anteroparietal sulcus here. And consistently with the literature, we also found visual areas. During the auditory exploration of the sound, it recruited, of course, auditory cortices, but also brain motor areas like the premotor cortex and the supplementary motor area of the cerebellum. And like for the haptic condition, we also find the visual areas activation during pure auditory stimuli. And finally, in the bimodal condition, you can see here that the network recruited is close to the union of the 2 unimodal networks. It's a strict combination of both. So by contrasting the bimodal condition with the 2 unimodal condition, we found that the superior temporal colliculus and S2 were specifically more activated during the bimodal condition, suggesting that this region may host audio-haptic interaction. However, based on this univariate analysis, it is not possible to determine whether this response increase is specifically related to texture processing. To answer this question, we did multivariate analysis, and here are the results. For the 2 unimodal decodings, that is the haptic decoding in blue and the audio decoding in yellow. Note that the decoding networks are subparts of the unimodal networks previously found in the univariate analysis, but without any visual areas activation. More precisely, you can see here the plot of the individual and mean prediction accuracy for the region where the accuracy was above 50% chance level. So for the haptic texture, it seems to be coded in S1, in the superior parietal lobule, SPL, in S2, of course, bilaterally, and also STS. For the auditory texture, it seems to be coded in STS, bilaterally, but also S2, and the left fusiform gyrus. The bimodal decoding points out the bilateral STS and S2 that were exactly the same region previously evidenced in the univariate contrast, suggesting that this region, also found in the univariate analysis, will process textual information. In conclusion, this study showed that haptic texture seems to be coded in S1, S2, SPL, and the STS. Auditory texture seems to be coded in S2, STS, and the left fusiform gyrus. But the posterior visual areas found activated during haptic and audio texture exploration will not be involved in texture encoding per se. And finally, STS-enhanced2 will be a heteromodal brain region hosting audio-haptic interaction related to texture information. I would like to thank you for your attention and to thank to all my collaborators. And do not forget to have a look on our poster. Thank you for your attention.
Thank you for organizing this session. I'm gonna talk about neurophysiological sources of multimodal neuroimaging in humans. The aim of this study is to investigate the extent to which the spatiotemporal dynamics of brain activity obtained by EEG or fMRI or the similarity-based EEG fMRI fusion are consistent with that of neural activity obtained by ECOG data. EEG and fMRI are two major non-invasive methods for imaging human brain function. EEG is known for its excellent temporal resolution but poor spatial resolution, whereas fMRI has a good spatial but poor temporal resolution. ECOG is an invasive technique providing simultaneous spatiotemporal mapping of neural responses. So it gives the possibility to compare neural dynamics with the results obtained by EEG fMRI fusion, but with the limitation of not having the whole brain coverage. In this study, we recorded EEG and fMRI data separately from 21 healthy volunteers. The stimulus set included 125 images from the animal, chairs, face, fruit, and vehicle categories. There are 5 identities in each category, and the identities are presented in 5 different sizes or orientations. This is the exact stimulus set that is used to record ECOG data by Liu and colleagues in the Kramer lab at the Harvard Medical School in 2009. The experimental parameters are also designed similar to that of ECOG data. In the following slides, I will present the results for the fMRI and ECOG comparison. To compare ECOG and fMRI data, we extracted brain visual regions in occipital inferior, fusiform, lingual, parahippocampal, temporal inferior, and pole temporal. Then, for each subject and each region, we extracted condition-specific voxel activity patterns. These are the inputs for SVM classifier to examine the category selectivity and the robustness to the scale and orientation changes in these regions. The results for ECOG and fMRI data are shown here. For each region, there are 5 bar charts, each showing the accuracy for classifying each category versus the all other categories. The bars are color-coded. For example, the blue bars are the accuracy for discriminating between the faces and all other categories. As shown in this figure, occipital inferior and fusiform areas are more selective to the faces than to the other categories in ECOG and fMRI. For parahippocampal and temporal inferior, fMRI and ECOG patterns of selectivity are different. fMRI responses show higher selectivity to the vehicles, whereas ECOG responses are more selective to the faces. To examine scale invariance, we trained the classifier with the images in the size of 3-degree visual angle and tested by the resized one. This is the average classification performance across different scales. We see that category information can be read out robust to the face changes in occipital inferior and fusiform in both fMRI and ECOG data. None of the lingual, parahippocampal, IT, and pole temporal regions show significant decoding accuracy across different scales in fMRI. However, in ECOG, these regions are more selective to the faces or fruits. A similar analysis was performed to investigate category selectivity under orientation changes. The classifier is trained using the images in the orientation of zero degree and tested by the rotated images. We observed that the classification performance is higher for faces in the fMRI and ECOG in fusiform area. In ECOG, the accuracies for faces and throats are almost identical in occipital inferior, whereas in fMRI, there is a significant difference between these categories' performances. In the next part, the results for ECOG and EEG data comparison will be presented. In order to evaluate the earliest time point at which we could reliably decode category information, we constructed condition-specific vectors using the EEG amplitudes over all electorals and used SVM classifier to decode object categories at each time point. In this figure, we see that the classification performance, as a function of time, averaged over all categories and subjects. The latencies and performance patterns for EEG and ECOG are compatible. According to this analysis, human brain decode category information as early as 100 milliseconds post-stimulus onset. The bar charts show the range of the classification performance for categories. For both modalities, the performance is higher for the face category. The third section is combining EEG and fMRI data and comparing the results with the ECOG results. For fMRI, in 6 aforementioned ROIs, we extracted condition-specific voxel activation values yielding 125 pattern vectors. Then we calculated the pairwise dissimilarity for all experimental conditions. The dissimilarity measures are assigned to a 125 by 125 fMRI dissimilarity matrix. Similar analysis was conducted on each data to obtain representational dissimilarity matrix at each time point. And then for each region, we correlated fMRI RDMs to EEG RDMs at each time point, and this gives signal over time. The first time point at which the correlation of the similarities is significant is comparable to the mean latency in ECOG data. There is a similar latency in occipital inferior, fusiform, and lingual areas, but not in parahippocampal, temporal, inferior, and pole temporal. In some, spatial analysis showed a great consistency between ECOG and fMRI results in occipital inferior and fusiform areas according to the time domain analysis, and regardless of the location, human brain can decode category information after 100 millisecond stimulus presentation. There is more temporal similarity between EEG and ECOG patterns compared to the spatial similarity between fMRI and ECOG.